So I'm delighted today to introduce Dr. Maria Fedorova, who comes from the Faculty of Medicine Carl Gustav Karras of the Technical University of Dresden. Just first of all, to apologize if I sound a bit quieter than normal, it's because I'm on day 3 of COVID, so my brain is in a bit of a fried state. But I am still delighted to be here, and I'm sure we're going to hear a fantastic talk from Maria. I'm really looking forward to this. So my name is Valerie O'Donnell. I'm based at Cardiff University in the UK. As normal, we're recording the webinar and it'll be uploaded onto Lipid Maps for those of you who can't make it. Around 45 minutes, and we'll have about 10 minutes at the end for questions. So what you need to do, if you think of a question during the talk, please post it in the chat or put it in the Q&A. And what we'll do at the end is we will unmute you and you will ask the question yourself so you can speak directly to Maria. So remember to post your questions in the chat because I'll generally take them in the order they're posted. And we're grateful to Cayman Chemical, sponsors of today's webinar. Thank you very much for your ongoing support. And our next webinar is open for registration. That's Alan Sagatelian, who's going to talk on lipidomics, approaches to find bioactive lipids on the 15th of March. So now to introduce Maria, it's my great pleasure to introduce Maria. I'm a really big fan of her work. It crosses over in some ways with our own interest in oxidized lipids. And I think, the work that Maria does, particularly on the methodologies, the software, and the discovery of structures of these lipids is really exemplary. And I'm really looking forward to hearing more in her talk. So Maria is currently research group leader in lipid metabolism analysis and integration at the Center of Membrane Biochemistry and Lipid Research, as I said, at the Technical University of Dresden. She studied biochemistry at St. Petersburg State University in Russia and obtained a PhD in chemistry and mineralogy in Leipzig University. And later on, she became a group lead for a number of years at the Institute of Bioanalytical Chemistry at the Faculty of Medicine and Mineralogy at the University of Leipzig. She moved not so long ago in August 2021 to the Center for Membrane Biochemistry and Lipid Research in Dresden, where she's now group lead. Maria's research is focused on development and optimization of chromatography and mass spectrometry methods for analysis of lipids in their modified forms. I mean, we all know as we move into, the increasing complexity of lipidomics, how important it is to have methods that we can use, that we can trust, and that are robust and rigorous. And, I think Maria's work is a really, really good example of, making sure that we use lipidomics correctly and, ensuring a really high standard in the work that's being done in this area. So she particularly works on implementation of high throughput LC-MS methods in discovery lipidomics. This is designed at targeting in-depth identification and quantification of the human lipidome in a variety of tissues, including particularly oxidized lipids, as she's going to talk about today under the term epilipidomes. By combining lipidomics data with investigation of related proteins and protein post-translational modifications using systems medicine approaches, she aims for further, deeper understanding of the pathophysiology of human disease, particularly focusing on obesity, insulin resistance, type 2 diabetes, and cardiovascular disease. And another of her focus of her research is development and application of approaches for analysis of protein and lipid modifications, obviously required for this research. And she's currently working to understand the role of redox lipid and protein signaling in the onset and progression of human disorders associated with chronic inflammation. Many of you probably will know that Maria is also one of the co-leads of the EU consortium EpilipidNet with Rosario Dominguez from Portugal. This consortium is providing fantastic opportunities across the EU and beyond, including us countries who've recently left the EU, in order to bring European lipidomics researchers together, further our understanding of the methodologies and the science, and really develop networks and collaborations. And I just want to congratulate her on her excellent co-leadership of the EpilipidNet. And also, I'm delighted to say she's working with us at Lipid Maps quite a lot, including implementing some of the excellent software that she has with her group, particularly NE, to analyze lipids and deal with some of the complexities of the large data sets that we're working with these days. So I look forward to continuing that work with you, Maria, and looking forward very much to hearing your talk today.
Thank you very much, Valerie, for this nice introduction. And thank you for inviting me to talk to all of you today. And like Valerie mentioned already, the focus of my talk today would be modified lipids. And I thought we will start with actually introducing the term epilipidome for those of you who doesn't know that. so you know what I will be talking about. So we all know that lipids, as a class of biomolecules, are extremely diverse. We have different types of head groups, and all those phospholipids, for instance. We have different types of backbones, glycerols, sphingosine bases, and we have the whole variety of fatty acid chains. And the combination of all those different chemical groups and moieties creates this lipid universe, as we know now, with thousands and maybe hundreds of thousands of species. But lipids also can undergo modifications via introduction of small chemical groups via enzymatic or non-enzymatic reactions. Just to give you a few examples, and I guess you know some of them already. Here we have a typical lipid, PAPE, which has a ethanolamine head group and two fatty acid chains. Like usual, one would be saturated palmitic acid, and the second one would be arachidonic acid, unsaturated one. So this lipid can undergo a whole variety of different modifications. For instance, it can get oxygenated or oxidized via action of lipoxygenases or free radical driven hydroxylation, hydroperoxidation, with the formation of the hydroperoxide group, for instance, at position 15, forming a new type of lipid, which is a 15-hydroperoxy eicosanoic acid containing one. So this lipid, you can imagine, has already different physicochemical properties and thus different functionalities in this, for instance, lipid membranes. This lipid can also undergo modification via non-enzymatic glycation in the reaction with reducing sugars, such as, for instance, glucose, forming a glycated form of this ethanolamine lipid. And that can happen, for instance, in a bloodstream if we have elevated levels of glucose, for instance, in conditions of type 2 diabetes or insulin resistance in general. And this glycated lipid with this modified head group with removal of the charge, obviously again have a new physical chemical properties and perhaps induce completely different signaling pathways. So with this in mind, a few years ago, we made a concept or named this concept of epilipidome as a subset of natural lipidome formed by lipid modification via enzymatic or non-enzymatic reaction required to regulate complex biological function. If you think about that, that's nothing really new in nature. We have already known a lot about, for instance, epigenomic regulation, when the methylation of DNA actually actively participating in different regulatory events in the cells. So it's again a small modifications introduced on the biomolecule, which without requiring a new biosynthesis of this biomolecule actually bringing a new function. All of you, of course, know a lot about protein post-translational modification with those different proteoforms of the same protein forming a new signaling pathways and regulating a lot of biological functions. You have protein phosphorylation. Of course, everybody knows in how many signaling events it's involved. So again, introduction of small chemical groups via enzymatic reaction in this case leads to the new functionalities in the protein without synthesis of the molecule de novo. So we can think about lipid modification or this lipid PTMs, if you wish, as this another level of complexity, which brings to the lipidome a certain gain in the regulatory capacity in regulating biological processes. And of course, there are different examples of such kind of regulations in the lipid universe exist as well. So one of the well-known examples is, for instance, regulation of different signaling functions over oxylipins. So we have arachidonic acid, which under the action of different enzymes like COX-2 and then different prostaglandin synthases can result in the whole set of different prostaglandins. Here you can see PGE2. And PGE2, for instance, have different receptors. There are 4 receptors expressed in different tissue in different cells. And that usually results in the activation, for instance, of macrophages, so pro-inflammatory signaling in certain conditions. Another example of a well-known lipid modification is, for instance, the phosphoinositides. So we know in this class of lipids, the modification of inositol moiety in the head group of those lipids with one to 3 phosphor groups in different positions, each of them carrying a certain functionality and certain signaling role. So one more example here, it's oxysterols. So oxysterols are a group of molecules formed by oxidation of cholesterol, and that happens either enzymatically on the different positions shown here, but also can happen non-enzymatically with certain preferences guided by the chemistry of this molecule shown here. And for oxysterols, there are also a lot of different functions assigned, depending on the position of hydroxy group introduced into the structure. So examples of lipid modification are well known around, and with those examples, we start to understand the whole functionality and this regulatory complexity induced by this epilipidomic regulation. So in an example of oxysterols, you could see that this modification and especially oxidation, which I am particularly interested in, can happen in different ways. That can happen enzymatically, for instance, via action of lipoxygenases, and you could see here the pathway, or it can happen by the action of free radical-driven reactions. But one way or another, the formation of the products might be pretty similar. But if we talk about enzymatic lipid oxygenation, those oxidized lipids are formed in very specific positional stereo and regio isomers. So there are different enzymes which can do that with a high specificity, and the products which are formed in these reactions are clearly defined in their stereo and regio chemistry and the positions of the modifications. Those processes are tightly regulated by the expression of certain enzymes. So the expression levels in different tissue and cell types, their localization, and the products are usually recognized by specific receptors. The free radical-driven peroxidation are usually used to be considered as something very unspecific and non-regulated process. But you can imagine that in cells, in the physiological conditions, the amount of radicals which can induce this kind of reactions is also tightly regulated. So if we talk about non-enzymatic oxidation, those oxidized lipids can be formed in the reaction with a free radical, such as reactive oxygen species or reactive nitrogen species, and that would result usually in the mixture of isomers. So this lipid peroxidation, as much as it seems to be random and non-specific, in the physiological conditions, it's still tightly regulated because we have a controlled concentration of free radicals in the cells. So those productions are usually governed also by different enzymes. And additionally, we have a very strong detoxification system present. If you want, you can call it a lipid quality control system on the level of lipid peroxidation here. So there is a whole number of radical-trapping antioxidants, as we know now, and they are produced so the level of those antioxidants can be increased or decreased by the action again of specific enzymes responsible for their production or renewal. And there is also different enzymes which can act on the level of lipid peroxide, actually bringing them to less toxic alcohol. And the best example which we know now with the development of the research in peroptosis field is TPX4. So we actually can think about this free radical-driven reactions as something which can happen as a fast way of adaptation to the environmental changes. So again, here we do not need to express new enzymes. We do not need to wait for that, but through these reactions, this modified lipid can exist. And that might be a mechanism in the redox sensing within the cells in the physiological conditions. So in addition to those oxylipins, which I mentioned before, we know that the free fatty acids in the cells, again, do not present in the high quantities, so they are quite, they have a detergent-like properties in cells, usually do not like to keep them free. So this kind of oxidation via free radical-driven reaction, they usually would happen to the complex lipids. So for instance, oxygenated phospholipids, oxidized cholesterol esters, or triacylglycerol. And when those lipids are getting modified, they would have different kind of modes of action in comparison to oxylipins. So what they can do in principle, yeah, so they can have some low affinity interaction with proteins. And obviously, if we modify lipids in this way within the membranes, we will have certain events leading to the membrane remodeling due to the changes of physical chemical properties of those constituents. So this remodeling can lead to the changes in polarities and electronegativities, and of course, changing the properties and activities of membrane-bound properties. So what else we know about the modes of action for those oxidized lipids is that actually, due to the changes in the membrane structure, those oxidized lipids, due to their higher polarity, yeah, so they can get out of the membrane bilayer, and this is usually called a lipid whisker model. So they can show up on the surface of the cells in the certain conditions, or for instance on lipoproteins. And we know now that those lipids can be recognized, for instance, by pattern recognition receptors and acting as a damage signal, and damage-associated molecular patterns, and leading to the activation of innate immune response. So there are different ways how these lipids can act, and that is actually one of my main interests in my research, to figure out actually which lipids are present there, and if we know what is there, we can look into the modes of action in the particular biological system, what is happening. And I would like to tell today you two stories. And the first one is pretty relatively old, but this is the way how I got really much interested in this complex oxidized lipids, and that's why I actually think it's important to look for these complex structures in different settings. So a few years ago, in one of my first projects as a PI, we looked in the dynamic model of mild oxidative stress. So for that, we used red cardiac myocytes, and we treated it with SIN1 with relatively low dose, and SIN1 is a donor of peroxynitrite, which can induce this mild nitrooxidative stress. And when we built up this model, so we took rather unusual time points to see what happens in the cell. So we looked at the cell after 15, 30, and 70 minutes upon the treatment. So we wanted to see how cell respond to this mild nitrooxidative stress in the time points when the expression of new genes and proteins is not happening yet. So what is the buffering capacity of this cell as a biological system itself to deal with this mild oxidative stress? As the first readout in the system to see what actually happens and if we have any oxidation, we looked in the biomolecular carbonylation. So this is such a general term which is describing introduction of carbonyl moieties in different biomolecules in the action of reactive oxygen species. And that happens pretty much to every type of biomolecules. For instance, for the DNA, this guanine is a marker of oxidative stress. It contains this carbonyl function. With proteins, can happen to a lot of modifications, which are leading to the so-called protein carbonylation and direct oxidation. For instance, deamidation of lysine with the formation of aminoadipic semialdehyde, or a reaction between nucleophilic amino acid residues in proteins with the products of lipid peroxidation via Michael addition, for instance. And lipids can also get carbonylated with the formation of such well-known markers of lipid peroxidation like malondialdehyde or hydroxymonoate. So we use this readout to see what happens in our model, and if we can see that we induce the stress and how cells react to that. In order to follow this carbonylation as a total event, we used a staining, which we developed also in my group using this coumarin hydrazide. I will tell later a few words about that. And this is a dye which can covalently react with the carbonylated moieties. But on the other side, it's pretty nice for microscopy visualization because it's cell permeable and we don't need to make any intensive pretreatment. So when we looked on this, let's say, total biomolecular carbonylation over the time scale of our model, we could see that very fast, already in 15 minutes upon the stress induction, we start to see increase of this carbonylation signal. It's going up over 70 minutes. And then if we leave our cells as it is, do not do anything else to them, 16 hours later, upon stress induction, stress are totally fine. They recover. So whatever they do, they manage 16 hours later to be all right. So total carbonylation, of course, can include carbonylation of DNA, proteins, and lipids. And okay, DNA, it was not the case because we didn't see the stain in the nucleus, but then we had the proteins or lipids. So it was easy to look for the protein carbonylation on the total level. There are Western blot methods which allow us to do that, where we derivatize those proteins with dimethylphenylhydrazine and then use the antibody against dimethylphenyl to see this carbonylation. So we could do this as well. And then when we compared the slopes of this curve and this curve, we could see that actually protein carbonylation is picking up later in comparison to the total carbonylation. So that means this increase in the first time points post stress induction is actually coming not from the proteins, but rather from lipids, since we excluded the DNA. So then we decided to look for this lipid-derived carbonyls. And of course, in the beginning, we didn't dare to dig into this complex lipids. So we looked for some well-known markers. And this would be this low molecular weight carbonyls, such as hydroxynonenal or malondialdehyde. Mass spectrometry, and as Valerie mentioned, I'm always interested to develop the new methods and look deep into the structures. So those lipid carbonyls are not so easy to analyze. So the small carbonyls, they're usually volatile, so you can lose them during your sample preparation, low molecular weight, and not easy to measure. So we use again this coumarin hydrazide, which I showed you before. And this is the structure of the molecule. And this is a really nice dye or reagent because, first of all, it allows you to link the carbonyls through this covalent reaction here. So give you a mass shift for the mass spectrometry measurement. Then we have this coumarin moiety. I showed we can use it for the fluorescent microscopy, but it also brings hydrophobicity to this adduct, so we can extract very efficiently this low molecular weight carbonyls and different hydrophobic solvents. And additionally, it has a steatorylamine group, which, as you can imagine, is a perfect group for ionization. So we can derivatize, stabilize our products, bring them all together in the extraction, and we also can increase the ionization, so the sensitivity. The other interesting thing about this CHH, what we call, is due to the fragmentation mechanism of those adducts, it actually allows pretty nicely deduce the whole structure of your derivatized products. Here is an example of hydroxynonenal derivatized with this dye. And you can just see that by this kind of marks that we are getting informative fragments in the tandem mass spectrum, which allows actually to define the whole structure of our aldehyde in this case. So we brought this method for LCMS application, where we can separate this alkanals, alkanals, and trienals based on their lengths of the chains. And in this model of our mild oxidative stress, we could quantify them relatively, and we could see that indeed these lipid peroxidation products, they are really picking up in the 30 minutes and starting to grow from 15, and actually the most abundant in the 30 minutes upon the induction of oxidative stress. Not all of them, but some, let's say, a very famous one, obviously going up very much, like hydroxynonenal here. Or in this case, for instance, we see oxabutanol methyl glyoxide. So they're really increasing very fast upon the stress induction. And then later on, they're going down. So the question, what happens with them? It's obviously they start to react with the proteins and start to modify proteins. And that explains why this protein carbonylation is actually getting much later up. So we looked for the protein carbonylation. I will show just one slide on that and not go into much in details since we talked today about lipids. So we also used the mass spectrometry-based approaches, derivatizing them with another tech, so which also can react with the carbonyl group in the proteins, and it has a binding moiety, so we can enrich them by affinity chromatography and then measure by mass spectrometry. And in this particular system, we got a high enrichment of proteins, modified those carbonylated proteins in the calcium signaling pathway, which makes a lot of sense for cardiomyocytes. And you can see here a representation of KEC pathway for the calcium signaling. So many proteins were modified, and we can even provide some functional readouts for selected ones. So we looked for the calcium signalings with the different receptors or channels, and we can see quite different responses. Yeah, so all of them were modified by this carbonylated lipids, and but some of them actually increased their activity, some decreased, and some stayed as it is. But the story with this model is interesting because, as I mentioned to you, that we see with this mild oxidative stress, which is inducing the cells, that we have oxidation events, something happens. But as I told you before, if we leave cells as it is, don't do anything to them, they will be able to recover. And we wanted to know actually what leads, which machinery is responsible for the recovery. So we look for the turnover of those modified biomolecules, and we started with the proteasome, yeah, because and that's one of the machinery which removed modified proteins in particular. And we could not see much going on here. So we see that, yeah, there is a little bit increase in the activity in ATP independent activity of the proteasome. So this 20S coproteasome in this one, but other way, it's not really changing. Here we use some inhibitor of proteasomal activity, and we don't see any increase in the carbonylation. Yeah, so here we see the total carbonylation in comparison to control. So yeah, it seems like it's not really influencing that. So then we looked for the turnover by autophagy and lysosomal degradation, and here we start to see some difference. So if we use this very general inhibitor, so MG132 inhibits both proteasome and lysosomal degradation, we start to see some differences, and even if we are using some more specific inhibitors for the lysosomal degradation. So then we looked in the autophagy induction in this particular system, and we could see indeed that already 15 minutes after induction of this oxidative stress, we have a very strong activation of autophagy. So, and that grows up pretty much over the time points. So additionally, what we saw here, that in parallel with activation of autophagy induction, yeah, we also started to see some structures accumulating in our cells, which stained very positively with Nile red. Yeah, so we can think about that, it's a lipid droplets. And additionally, they stained positively with antibody against oxidized phosphatidylcholins. Yeah, there is this antibody available commercially, EO6 antibody, which can recognize the head groups of oxidatively modified phosphatidylcholine lipids. And it's also stained positively with coumarin hydrazide, which we see. So what happens? This is again the staining with this EO6 antibody. So if we treat our cells with SIM1, induce this mild oxidative stress, cells start to accumulate lipid droplets, and that is coincide with activation of autophagy. And those lipid droplets are presumably filled with oxidized lipids. If we inhibit the lysosomal degradation here, this process is not resolved by the cell. Yeah, so here you could see if we leave them alone, they're able to manage that. But if we start to interfere with lysosomal degradation, then this process is going on and cells cannot manage anymore this accumulation of oxidized lipids. So we could see that this kind of lipid signaling, this action of modified lipids in the first moments of the stress are very important, and this autophagy lysosomal degradation is really critical to manage this kind of clearance of modified biomolecules. So we looked further on into this pathway by looking in more specific with more specific inhibitors, yeah, for instance, of autosome formation, of the fusion, and lysosomal degradation as well. And there was also a lot of interesting results in that. So here you could see some staining again for the CHH, carbonylated lipids, Nile red for lipid droplets, and again we see these nice colocalizations. So I'm not going into details, and if you're interested, you can read the paper about that. But the main conclusion here, which we can say, is that if we look at our cellular system here and start to induce this mild nitrooxidative stress, it's very so lipid peroxidation or lipid oxidation come first, and that this lipid oxidized lipids can modify proteins that will affect the functions. But if autophagy lysosomal degradation is activated here, we can get rid of them and so on. So that really raised my interest in trying to understand what is actually happens here so fast, where those oxidized lipids coming from. Is it a random distribution of any lipid around, or there is any specificity for any particular sites, intracellular membranes, and so on, and how efficiently this formation of lipid droplets can eliminate those modified lipids, and all this kind of interplays. But what we can understand already from the study, if we look into these oxidized lipids, by using this small products such as hydroxynonenal MDA, that doesn't help us to understand where this process actually starts and which membranes come first and so on, because this kind of products, small aldehydes, can be formed from any kind of lipid pretty much, which has polyunsaturated fatty acids. So we really need to look into the ones which are those complex oxidized lipids in order to understand where they're coming from and what is happening there. So over several years, we looked for many different types of modified lipids and trying to address these analytical challenges. So we have this low abundance in biological systems. So that would require enrichment or targeted mass spectrometry. But from the other hand side, we have this very high chemical diversity. We have so many unknown lipids there, so that without considering this biological context, we have no chance to find out what is out there. And we developed a number of methods with this epilipidomic platform, for instance, for oxysterols, for head group modified lipids, oxidized nitrated fatty acids, this low molecular weight aldehydes, which I showed you today, some protein adducts. But now the main interest here is in the modification of complex oxidized lipids, because knowing which lipids is actually getting oxidized, we can estimate from which kind of membranes at least it comes, or which kind of groups and subclasses. So with that, I come to the second part, and what we did recently over several last years, where we tried to develop the methods which allows us to measure this complex oxidized lipids and address this diversity of the epilipidomes. So from mass spectrometry point of view, doing the work on this complex oxidized lipids was relatively complicated, because if you worked in the analytical biochemistry or bioanalytical chemistry, so if you want to develop a method to measure something, you usually start from the standards. So you buy standards, and then you develop your method, your LC separation, for instance, your mass spectrometry detection, your MSMS settings, using those standards which would represent those lipids, for instance, in this case. But for oxidized lipids, there are not so much standards available, and they are far from being representative for the diversity of these different chemical groups which can be present there. So what we did here, we performed a lot of experiments where we oxidized phospholipids and neutrolipids in vitro and studied their fragmentation and mass spectrometry behavior. We also collected all possible information out there on the literature on the different fragmentation patterns which one can find. So different databases, everything that's available also on lipid maps, MS-dial library, and so on and so forth. So combination of this kind of in vitro oxidations and also study of the all available literature allows us to formulate these fragmentation patterns and rules. So we know now how we can assign the fragmentation patterns, for instance, for modified phospholipids. And we can see the fragmentation of the main events, which allows us to identify the lipid class, the type of the modification based on the neutral losses, but we also looked very deeply into the different fragments, which allows us to distinguish different modification sites, so the isomers. And we could define the set of fragments for each, for instance, position of hydroxy, hydroperoxy, or keta groups introduced into that cell chain. So we also understood that application of high-resolution mass spectrometry and the MS2 dimension here is also crucial, because in this list of different fragments, we also could see a lot of isobaric, closely isobaric fragments. Yeah, so we need also to have a high-resolution mass spec for the MS2 dimension. So then we also, so the isomeric or isobaric species are formed a lot within oxidized lipids. So we heavily rely here on the application of a liquid chromatography in order to separate those isomers. So you see here the signals, so the peak solution profile for different oxidized cholesterol esters. In one color, you could see the isomeric species, for instance, double oxygen addition on this CE represented by multiple species, and using also standards and also standards of oxylipins, let's say, we could define the elution rules also for those kind of isomeric species, which are shown here, for instance. And we use it a lot by plotting, for instance, those retention times versus Kendrick mass defect, because this was shown to be a very useful tool to actually align those structure and to find, for instance, some artifacts. Yeah? So, for instance, in source fragmentation is a very strong event in lipids in general, and in oxidized lipids in particular. And for instance, the hydroperoxides, independent on the class, is it for CE, TG, or PC, they tend to in source fragment into the corresponding carbonyls. And if you do not know that, you can actually misidentify those species. And you could see here that with this Kendrick mass defects, you distinguish this artificial in source fragments for the real from the real oxiderivative quite easily. So the other thing which we did into optimizing this mass spectrometry-based solutions here for oxidized lipids is finding optimal ionization adapts. So what we notice that can change upon lipid is getting oxidized. So CE, for instance, or TG, usually detected as ammoniated adduct in the positive mode. But we notice that when we deal with oxidized CE and TG to this matter, they actually tend to form sodiated species. Now you could see here extracted ion chromatogram for the same oxidized TG, represented as in blue as ammoniated and in green as a sodiated adduct. So first we saw that would be a problem, but it turned out to be actually a nice feature, because those sodiated species resulted in much more informative MSMS. If you look on the ammoniated adduct, here it's a classical fragmentation pattern for instance, CE. Yeah, so the only thing which you actually see is a ion of holocene here. But if we look on the MSMS of those sodiated ones, we start to see much more features or fragments which help us to identify the structure. Those sodiated products were more resistant to the fragmentation than the ammoniated ones, so we had to work a bit on our collision energies. But on the other hand side, it's also better for reducing in-source fragmentation, since they are more stable, they also less in-source fragment. And with all those optimization, we can up with the methods where we can use just MS2 without need for MS3. And we can get a very informative spectrum, which allows to define the structure of oxidized cholesterol esters, oxidized TGs, and also oxidized phospholipids, but I do not show it here. So oxidized phospholipids we measure in the next defined mode. So with all this information, we have now a method which allows us to measure oxidized lipids accurately. So we know their mass spectrometry behavior, MS2 behavior, we know their elution patterns, and so on. But that still doesn't help us to address this diversity of oxidized lipids. So we have a method sensitive enough, specific enough, but we still don't know for which lipids to look in the given biological system. So here we can talk about this lipidome oxidizability. So that the point is that epilipidome composition in a given biological system will be determined by different factors. The first factor is obviously substrate availability, so membrane lipids, for instance, and their puffer content. You can imagine that lipidome of different tissues and cells is different. That means there is a different substrate for the formation of oxidized lipids. We know that membranes are asymmetric. We have, for instance, on the upper leaflet, a lot of more saturated PC and sphingomyelins. On the inner leaflet, we have a lot of plasmalogens and PEs, which have a lot of puffers. So even the two different leaflets will have different, let's say, substrates for the formation of epilipidome. Of course, the mediation of radical propagation is important, the detoxification system, which I mentioned before. And in principle, every step in this kind of equation, the formation of initiation, propagation, and termination would influence this epilipidome composition. So how to deal with this large sample-specific diversity, but on the other hand, this low abundance of oxidized lipids in natural lipidome. So we want to measure in the condition, not when system undergo a massive oxidative stress. We want to measure in the physiological or pathological conditions where subjects are still alive, obviously. So how we can account for all those different steps? Obviously, now we cannot do that for all of those steps, but what we can do, we can account for the substrate specificity. So with this, we came to this concept of sample-specific epilipidomics, and that is what we do now in the lab. So when we want to measure epilipidome of any given biological system, the first thing which we do, we measure a native lipidome. Yeah, so we do a regular lipidomics where we identify lipids or lipidome of this particular cell type, this tissue or blood plasma. And then we use this native lipidome to predict the sample-specific epilipidome. So for that, we use a software which was developed by me in my group. And this software, LIPID, allows us to perform identification of oxidized lipids from LC-MS data. But importantly for this workflow, it also allows us in silico oxidized any given lipidome and predict which oxidized lipids can be formed. And this prediction is based not on the simple enumeration with the oxygen atoms, but it's based on the reconstructed networks of polyunsaturated fatty acids oxidation. So Nii and Georgia back then did a lot of work on collecting a lot of literature on this kind of reactions, which have ever been shown to happen to 10 most abundant puffers. And then Nii implemented these networks in this in silico oxidation algorithm within LIPID. So we measure native lipidome and use it for the oxidation. And then we can go with a semi-targeted LC-MS analysis using this predicted epilipidome for identifying what is actually present in this biological system. Then, of course, we can use LIPID for matching the spectra and identify that. So these reconstructed networks were really instrumental, and this is shown only for one fatty acid. I think it's cholinolenic acid, but in the reality, the whole network looks like that, where we have more than 500 reactions for more than 480 different oxidized derivatives. So, and at the end, I will show you just one example what we recently did with this approach. So we used this approach on the real samples, and in this case, we worked with the blood plasma sample from the obese and lean individuals. We had 50 samples from lean individuals, we had 50 samples from obese but insulin sensitive, and 50 samples from obese but insulin resistant. So we know the composition of blood plasma, of human blood plasma lipidome. And we took this composition and used, for instance, 17 PUFA-containing PC, 17 or 11 CEs or 17 TGs containing PUFA, and use those one to predict possible modified lipidome of the human blood plasma. So you could see here that the prediction was quite huge. Yeah, so we have around 1,000 of possible oxidized species formed from those 45 unoxidized ones. So we used those 1,000 lipids or MOZs for semi-targeted analysis. So we use it as an inclusion list for the data-dependent acquisition, and then went through this all identification steps and validation steps, which I described to you. So with this approach, we could identify a half of those lipids from, or sorry, half, 1/3 of those lipids, which we predicted to be present actually in those biological samples. And then when we know what is present there, then it's pretty easy and straightforward. So we can perform targeted quantification using, in this case, PRM on the individual samples and perform statistical analysis. So we could quantify over 100 lipids here, oxidized lipids, and of them, more than 70 showed a significant difference between these different groups. This is how that looks like again. So we take those unmodified lipids containing polyunsaturated fatty acids from a given lipidome, use LPP Tiger to predict oxidized lipidome, and then used in the pooled samples, group-specific pooled samples, we use this semi-targeted data-dependent acquisition to look for those predicted lipids, and we do it with the polarity switching. So for instance, oxidized phospholipids we measure in the negative mode, oxidized CE and TG in the positive mode. That also saves the time and give us, so we can use less samples here. And in one LCMS analysis, you can cover pretty much. So then we go through all this identification procedure using our optimized settings, parameters, fragmentation patterns, and elution profiles. And that works pretty nice because we were able to go on with this workflow and then from the identified species to the targeted acquisition by using only 1.6 microliters of blood plasma. So finally, what did we get from that? So we could identify and relatively quantify the differences between the three different groups of patients. And we did it for oxidized phospholipids, so phosphorylcholine, CE, and TG. So when we looked at the data, and maybe we had an idea that we will see, like black and white, so no oxidation in lean individuals and a lot of oxidation in obese individuals, but that was not the case. So we saw here that in lean individuals, so this is the last row, and you see here a heat map which is showing the intensities of those modified lipids in lean, obese, insulin sensitive, and obese with type 2 diabetes. So we can see that already in lean individuals, we see a group of oxidized phosphatidylcholines which are present in much higher quantities relative to the obese ones. And there is another group of oxidized phospholipids which is present in a higher quantities in obese individuals. So there's a clear separation between lean individuals and obese ones in the pattern of their oxidized phosphatidylcholines. When we start to look closely on that, so let's say on those lipids which are present in the lean individuals, let's say physiological level, as much as we can say that, we could see that all of them have a 18 carbon long modified chains. So all of those oxidized lipids here are octadecanoids. And their levels were not that high because we could identify that they're oxidized, but rarely we can distinguish the type of modification or the position. And when we look now on the obese individuals, we see that majority of the modified species here are carrying eicosanoids, so acyl chain with 20 carbons. And you could see there is a lot of peroxides here. And we also have hydroxide with different positional isomers And if you see in these positions one, so we can say, for instance, position 9 is usually not formed enzymatically. So this looks like we have increased level of lipid peroxidation and formation of all those species here. So this physiological pattern of octadecanoids present in lean individuals is completely scrambled with the development of obesity, and looks like we have intensive lipid peroxidation going on, mostly on the acyl chains, these 20 carbons. So interestingly, this pattern was very reproducible in the other classes. So in TGs and CEs, again, we see the group of modified lipids which are carrying modified acyl chain with 18 carbons. And then when we move to the obese individuals, we have mostly, again, here, I think the most clear with CEs. So again, longer chains, 20, even 22, with a whole variety of different products. Many of them are hydroperoxide as well. So then in all those different classes of lipids, so phosphatidylcholines, triacylglycerols, and cholesterol esters, this let's say physiological, if we can call it, pattern, characterized by the presence of modified octadecanoids, and that is completely scrambled with the development of obesity. So that stands to the formation of lipid peroxidation products, and mostly from the longer acyl chains. For instance, here it was also interesting if we look on the pattern of modified cholesterol esters in lean individuals, we see here mostly epoxide and diols. Yeah, and this is also point out that epoxide diols are usually classical products of cytochrome P450 driven oxidation, for instance, in liver. So maybe this is coming from the liver in the physiological condition and have certain functions. So with that, we could propose that those modified lipids in the physiological conditions might be carried along the bloodstream in the form of lipoproteins, and they probably have some physiological functions and might participate in the endocrine signaling. So maybe at certain conditions they are taken up via lipoprotein uptake or released by lipolysis and can actually execute their physiological functionality. On the other hand side, with the development of metabolic distress, complications, these physiological patterns is completely shifted, and we have formation of very different other species, which we also need to figure out what actually they do and how they can be released. So with that, it's open us in like a new perspectives to look on those modified lipids and to see how informative is that when we actually can assign the particular modifications to the lipid classes and their distributions to find these new regulatory patterns and how actually that is all changing with the development of metabolic complications. So with having this method in our hands now, we are also working a lot with the cell models. So try to understand what happens in the, let's say, when we induce oxidative stress, redox disbalance, ferroptosis, or whatever it is, and we can look what happens in the cell and try to find, based on the pattern of those oxidized lipids, if we have any particular membrane specificity, and we can allocate this event to different cellular compartments. So there is a lot to look into, and by having these tools nowadays, we are very optimistic and sure that we can find a lot of different pathways in the regulation of the epilipidome. So with that, I would like to conclude and thank people who participated in this work. So going back for the work with the cell models, so this is the mainly work of Eva and Venu. They are long time gone from my lab, but still. And for the oxidized complex lipids, so this, of course, the people who were very instrumental to that. This is Angela and Paulina for the lab-wet work, me with Lipid Hunter, and also Georgia for the networks and Dieppe as well. And thank you very much for your attention.
Thank you, Maria. That was excellent. We have a few questions, but I'm going to take chair's action and start, actually. One question I had about the last data that you showed, where you had this switch when the volunteers were obese. Is this extracellular vesicles coming from adipose tissue that's contributing those phospholipids with the arachidonate potentially? That it's different particles contributing rather than a change in the composition of the lipoproteins? Could that be what you're seeing?
Yeah, that's also could be. That's why we actually now want to look in the, let's say, different fractions, yeah, in the blood plasma or blood in general, right? So at least starting with a lipoprotein for sure. So because we see also the specificity of modification. So maybe if we look on those modified lipids, not in the whole plasma, but in the fractionated plasma, for instance, at least for the lipoproteins, yeah, we will get more mechanistic insight what is going on.
Yeah,
but it also can be the extra cell vesicles, yeah. So well,
from adipose tissues, and then going back to the biochemistry that you talked about earlier on in relation to the role of lysosomes and lysosomal degradation, I was quite interested in that because obviously if we're going to degrade oxidized phospholipids, we've probably got to break them up into their constituent parts first, and then you'd have beta oxidation of the oxidized fatty acid part, which is generally peroxisomes, mitochondria, etc. So what's going on in the lysosomes? Is that the lipase part, and then they get shunted to other organelles, do you think? Or what do you think is going on in lysosomes?
Yeah, I mean, lipase part is absolutely critical, yeah, there, because then when we, in the second study, which I didn't focus so much today, we also use this lipase inhibitors, right? And that has a drastic effect, obviously, on this process, yeah. So what happens then? This is also a question, yeah. So I think it would be interesting to look using the methods which you recently published, right, with this processing of oxidized fatty acids further on, so to look the degradation,
whether they get shuttled between different organelles. Yeah, that could be interesting. Okay, so a few questions. The first one, I'm going to ask you to unmute yourself. It's Incy Barrett. You can speak now.
Hello, thank you for your presentation. I would like to ask, how did you measure the proteasomal activity? Like I saw you measure the activity of proteasome, and I'm wondering how.
I will not tell you now all the details, but you can look it up in the paper and the method. There is all there. But the general idea, you are using a peptide, which is represent the site for, I think, for hematrypsin activity in that case, and it's fluorescently labeled. And when it's cleaved, you are getting, let's say, decrease of the fluorescence or increase. I don't remember that particular case what it was. And when you isolate, let's say, the proteasome preparation from your cell, so you can measure using this method, and depending if you add or not ATP additionally, you can access to 20S versus 26S proteasomal activity, yeah, because 26S requires ATP and 20S does not. And in our case, we saw that 20S is actually start to increase a bit. That makes sense because usually they say that 20S is the one which act on oxidized molecule proteins, yeah, because it's more robust and doesn't need so much energy. But for details, please look in the material methods. Everything is described there pretty good, I think.
Thank you.
Okay, thank you. So, Bryn Sundberg.
Great, thanks. That was really impressive. I was hoping you could talk a little bit more about the retention time and Kendrick mass defect filtering that you do and where you kind of place that in your process. Is that something that happens really far upstream and you can just automate it and do it for everything that you potentially identify? Or do you look at only a few statistically significant, and is this like a very manual process?
Yeah, so we became recently a very big fans of this Kendrick's mass defect, following on the paper from Ansgar when he not introduced, but at least brought back it to the lipidomics and oxidized lipids as well. So we use that in different ways. So we apply that as one of the first post-processing steps. So definitely before any significance and so on. to actually confirm our identifications. Yeah, so we go through the MSMS spectra with LPP Tiger and then with manual verification, and then we have a list of our identified lipids with corresponding retention times. So we know elemental composition, we know retention time, we know our identifications, yeah, what we assign to this one, and then we build this Kendrick mass defect plots. So there are some scripts which we have to do that. For doing that automatically, they are available on the GitHub page. So if you go to the paper, there is a link there to that one. Or you can also, they don't look so, let's say, beautiful if you want to use it for the publication and so on. So when we want just to have a better picture, then we rebuild it, I don't know, in Origin or something like that. But you can do it with these R scripts. And that is absolutely, I could not underline it more, instrumental for finding this in-source fragmentation and the pulse positive identification. Yeah, with all these isomeric species, there is, when you go to the complex lipidomes, this is something I would say really must.
Great, thank you so much.
So we had a question about software, but it has been answered in the chat by one of the other attendees. So I'll move on to Amir Ravandi. You should be able to talk now.
Thank you. That was a wonderful, wonderful talk. I had a quick question. Do you think it's a difference between lean and obese in terms of the availability of substrate and then hence the difference in oxidation process, or is it, do you think this specifically something different in the potential maybe reactive oxidant species that hits these compounds?
I think it might be both, but it definitely also has something to do with availability of substrates. Yeah, again, because for this data set on these plasmas from obese lean individuals, we also have a lipidomic data set, yeah, where we measured quantities of normal lipids. Yeah, so we can do some correlation, and there are correlation definitely with increasing oxidation with in obese individuals in obese individuals, we see, for instance, more oxidized CE with arachidonic acid, let's say, but we also see more CE with arachidonic acid in obese individuals in general. So for this obese complications, this correlation totally goes on. And for lean individuals, I would say it's something more specific because still, for instance, some of the lipids may be more abundant, but the octadecanoid ones are more present. Yeah, I hope I explained myself.
No, that was great. Maybe I can ask this another question. What about the carbonyl compounds? It's always hard to say what are bound, what are free, and just with regular lipid extraction, are we anywhere closer to say, are we getting a full understanding of how many oxidized lipids are actually there when we just do a regular lipid extract, or do you have any other ways to get around it?
I think as much as I look in the oxidized lipids, yeah, so it gets more and more complex. And the point is that what I'm sure very much now that it's really sample specific. You cannot apply the same set of, say, okay, look for the same lipids in different systems, yeah, because they will be different, because the lipidome of different system is different and it serves as a substrate for that. So that's why there will be no uniform set of oxidized lipids which you can measure in brain, in liver, in blood plasma, in some tissue extract. So there always will be specificity which is determined by the lipidome itself. Yep.
Okay.
But we need to find the patterns which will make sense.
Okay, so I think we have time for just one very quick last question from Mark Zatzi. You should be able to unmute now, Mark.
Hi, thank you so much for your talk. So my question was, in your data, you're showing data for mostly PC species. I just wanted to find out if the tendency for modification of phospholipids is similar in like different lipid classes as well.
Yeah, that also depends on the system in question. I must say in the plasma, in this set which I showed you now, we also look for PE, yeah, but not much happened there. Yeah, in plasma, at least, we didn't see many oxidized PE, and none of them was actually statistically significant. But in other systems, yeah, in cell systems, for instance, we see a lot of oxidized PE. So again, this is totally dependent on the biological system in question. So.
Thank you.
Okay, thank you, Maria. I think that's all the questions. That was a really excellent talk, and thank you for coming today. Thank you to all our attendees, and we look forward to seeing you in another few weeks for our next webinar. Thank you. Goodbye.
Thank you. Bye-bye.